Good day everyone, I am Kimberly Nicole Grana and my topic for this report is all about central dogma of molecular biology. So the central dogma of molecular biology states that DNA contains instructions for making a protein which are copied by RNA. So RNA then uses the instructions to make a protein. In short, DNA to RNA to protein or DNA to RNA to protein. So RNA then uses the instructions to make a protein. In short, DNA, RNA to protein or DNA to RNA to protein. So why is it called the central dogma of biology? These are protein to protein, protein to RNA, and above all, protein to DNA. This was that Crick meant when he said that once information has gone from DNA into the protein, it could not get out of the protein and go back into the genetic code. So the central dogma of molecular biology is an explanation of the flow of genetic information within a biological system. It is often stated as DNA makes RNA and RNA makes protein, the central dogma. This states that once information has passed into protein, it cannot get out again. So in more detail, the transfer of information from nucleic acid to nucleic acid or from nucleic acid to protein may be possible, but transfer from protein to protein or from protein to nucleic acid is impossible. So as you can see in the picture, the central dogma, which states the DNA to RNA and to protein. The dogma is a framework for understanding the transfer of sequence information between information-carrying biopolymers, in the most common of general case in living organism. There are three major classes of such biopolymers. The DNA and RNA are both nucleic acid and protein. So there are 3 times 3 is equal to 9 conceivable direct transfers of information that can occur between these. So the general transfer describes the normal flow of biological information. So the DNA can be copied to DNA or DNA replication. The DNA information can copy into mRNA or the transcription, and the proteins can be synthesized using the information in mRNA as a template or what we call translation. So the special transfers describe RNA being copied from RNA or RNA replication, DNA being synthesized using an RNA template or what we call the reverse transcription, and proteins being synthesized directly from a DNA template without the use of mRNA. So the central dogma of biology is best described by DNA is transcribed to RNA, which is translated to protein. So the genetic material or the DNA is transcribed into mRNA or RNA, which is then translated into proteins. Biological sequence of information. So the biopolymers that comprise DNA or RNA and polypeptides are linear monomer polymers. Examples: each monomer is connected to most two other monomers, so the sequence of their monomers effectively includes information. So when DNA is transcribed to RNA, its complement is paired to it. So the DNA codes is the A, G, T, and C are transferred to RNA codes U, C, A, and G respectively. So the encoding of the proteins is done in groups of 3 known as codons according to the table. So the other examples of central dogma is an analogy might be that central dogma is like making your mom's recipe for brownies. So first, you called your mom who represented DNA. Then, you listen and copy down for instructions. So this is like transcription because during transcription, DNA is copied to mRNA. So the DNA replications. In the sense that DNA replication must occur if genetic materials is to be provided for the progeny of any cell, whether somatic or reproductive. The copying from DNA to RNA arguably is the fundamental step in the central dogma. So a complex group of protein called the replicism performs the replication of the information from the parent strand to the complementary daughter strand. So the replicism comprises a helicase that unwinds the superhelix as well as the double-stranded DNA helix to create a replication fork. The SSB protein that binds opens the double-stranded DNA to prevent it from reassociating. So the RNA primase that adds a complementary RNA primer to each template strand as a starting point for replication. So DNA polymerase 3 that frees the existing template chain from its 3 and to its 5 and adds new complementary nucleotides from the 5 and to the 3 and to the daughter chain. So this DNA replication is the special transfer in the central dogma of molecular biology. It is one of the 3 processes of the central dogma. So the 3 processes, first is the DNA replication, the transcription, and the translation. So the transcription is the process by which the information contained in a section of DNA is replicated in the form of the newly assembled piece of messenger RNA or mRNA. So enzymes facilitating the process include RNA polymerase and the transcription factors. In eukaryotic cells, the primary transcript in the pre-mRNA. So the pre-mRNA must be processed for the translation to proceed. So the translation is when the mature mRNA finds its way to a ribosome, where it gets translated. So in the prokaryotic cells, which have no nuclear compartment, the processes of transcriptions and translation may be linked together without clear separation. So in eukaryotic cells, the site of transcription or the cell nucleus is usually separated from the site of translation or the in the cytoplasm. So the mature mRNA finds the way to be a ribosome, where it gets translated. So in the prokaryotic cells, which have no nuclear compartment, the processes of the transcription and translation may be linked together without clear separation. So in the eukaryotic cells, the site of the transcription is usually separated from the site of the cytoplasm. So the use of the term dogma. In his autobiography, What Mad Pursuit, Crick wrote about his choice and the word dogma and some of the problems it caused him. So I call this idea the central dogma for two reasons, I suspect. I had early used the obvious word hypothesis in the sequence hypothesis, and in addition, I wanted to suggest that this is the new assumption was more central and more powerful. So as it turned out, the use of the word dogma caused almost more trouble than it was worth. Many years later, Jacques Manonod pointed out to me that I did not appear to understand the correct use of the word dogma, which is a belief that cannot be adopted. I did apprehend this in the vague sort of way, but since I thought that all religious beliefs were about or without foundation, I used the word the way I myself thought about it, not as most of the world does, and simply applied to a grand hypothesis that, however plausible, had little direct experimental support. So if you were asking, does the central dogma state in the molecular biology and how it differs in some viruses? So very soon, Francis Crick proposed the central dogma in molecular biology, which states that the genetic information flows from DNA to RNA and to protein. In some viruses, from the example, the retro, a process called reverse central dogma is performed in which the RNA genome of the virus is processed into DNA. So before to end this slide, I just want you to add information. So the molecule of the mRNA then leaves the nucleus and goes to the ribosome and the cytoplasm, where the translation occurs. So during the translation, the genetic code in mRNA is read and used to make a polypeptide. So these two processes are summed up with the central dogma of biology, which is the DNA to the RNA and to protein. So again, the statement, as I'm going to mention, is the accurate statement to represent the central dogma. So the DNA is transcribed into RNA, which is then translated into protein. And the central dogma is a framework for understanding the flow of genetic information, since it states that DNA makes RNA and RNA makes protein. Transcription occurs inside the cell's nucleus, and then RNA leaves to go to the next step. The next step is called the translation, as what I mentioned earlier. Since the central dogma helps us understand the process by which DNA turned into protein, so the steps involved in the process in the gene expression is that it consists of the two major steps, which is the transcription and the translation. So together, transcription and translation are known as the gene expression. So during the process of transcription, the information stored in the gene's DNA is passed to a similar molecular called RNA or the ribonucleic acid. in the cell nucleus. Lastly, the central dogma instructions on the DNA are transcribed into messenger RNA. So the ribosomes are able to read the genetic information inscribed on the strand of the messenger RNA and use this information to string amino acids together in a protein. So my report is all about. Thank you for listening. Have a blessed good day to everyone.